Good morning, good afternoon, and good evening, everyone. And welcome to this Scientist.com webinar titled Integration of the HTG Transcriptome Panel into Preclinical and Clinical Programs to Drive Precision Medicine Research. I'm Liam Sanyo from the Scientist.com events team, and I'm pleased to be your host for today's event. This webinar has been sponsored by HTG Molecular, so a big thanks to them for helping to make this program possible. We're very fortunate to be joined today by Dr. Peter Ansell, Scientific Director and Research Fellow in Precision Medicine Oncology at Abbvie, and Dr. Marion Navratil, Senior Vice President of Research and Development at HTG. And today they're going to describe the use of novel RNA-based gene expression profiling technologies for clinical and preclinical oncology research. And with that, I'm very pleased to welcome the first speaker, Dr. Peter Ansell. Pete, thanks so much for joining us today, and the floor is yours whenever you're ready.
Great, thank you so much. Thank you for the introduction and for the opportunity to be here today. And thank you for everyone for joining. Really looking forward to having an open dialogue about basically what we all care to do, and that's take our research farther, faster. And so we'll walk through some of the work that we've been doing here at Abbvie using the HTG technology, so the folks can be familiar how we're applying it. And then Marion will walk through a little bit more about the nuts and bolts of the technology and just look forward to a great scientific exchange. So my interest, in research, coming as a cancer biologist and precision medicine researcher, really have a long-standing interest in using gene expression profile to really understand tumor biology, to segregate tumors along that biology with the hopes of understanding sort of drug response and to make better predictive tools to inform which patient should be treated with which drug during the course of clinical development and ultimately at commercialization and launch. And so we've done this throughout a number of clinical instances, and here's a few sort of examples that colleagues and I and collaborators have published. The top one on triple-negative breast cancer, sort of using gene expression profiling to associate with clinical outcomes there. That was done using traditional RNA-Seq approaches and assigning subtypes. And then the bottom two are in non-small cell lung cancer. Again, these are just examples, but where we look at understanding patients in combination with platinum and a PARP inhibitor in squamous non-small cell lung cancer. And there we applied, again, signatures, gene expression signatures to understand patient response. And then the final paper there, again, is in non-squamous non-small cell lung cancer. And this one is a little bit different than the previous two. The previous two used whole transcriptome-based RNA-Seq to do that in more of an exploratory fashion. The final one there was a phase 3 that we're trying to seek approval for a biomarker RNA-defined expression signature to enable registration. And so with that, the use of what would have been, ultimately a companion diagnostic to understand gene expression and how to faithfully categorize patients for drug response and then ultimately approval. And that one was used targeted assay using the HTG technology. So we have a wide variety of experience in different clinical settings, different disease settings, and different technologies to try to use RNA expression to understand patients who will or will not benefit from a given drug. And so through that, and certainly people on the call have faced these types of challenges. One, if we go to certain databases or we want to understand a disease, oftentimes DNA-based markers are used as commonplace, and RNA is not widely used or not routinely used. The other sort of feature that we find are sample size or quality. We're continually trying to get more and more answers out of smaller and smaller samples. And sometimes the samples, when we run these worldwide studies and we get patient samples in, the quality can be variable. And so we need to work with technologies that can really engender results high quality results with samples that are not optimal. The other thing, especially in drug development, long turnaround times. If we're trying to make decisions nimbly during the course of early clinical development, we oftentimes enroll patients, wait a month, understand safety signals, response signals. We want to have those biomarker results within that same time frame. And so, technologies or situations where if we need to wait, 2-3 months for results, it's not as helpful as it could be. The other situation we see are experimental biases from many different types of technologies, sources, and could be from sample sectioning. And if we want to compare one biomarker to the other, and they're done on serial sections or different parts of the block, some extractions of nucleic acids are better than others, and can all cause bias in your experiment. The other thing is, of course, processing unconventional sample types is certainly a challenge. Most of what we do is FFPE, but that's also not easy to work with, and there are other sample types that need to be dealt with. And then, finally, deciphering sequencing data. Which pipeline to use, running pipeline. The sequencing is one thing, but then if the informatics on the back end are challenging and lengthy, that also adds to the experimental time and takes away from what we really want to be doing, and that is, turning those data into knowledge to move programs forward. So these are sort of the challenges that we've encountered, and we think we've made some good progress that we'll talk to a little bit today. So let's walk through, I guess, again, what our experience has been with the HTG technology, and then Miriam will go into more detail about what it is and what it's not specifically, so that we can all increase our awareness. So as I alluded to in the introduction, we've been using gene expression profiling, in many different instances, and including using the HTG, a targeted panel, in a registration enabling study. So we're somewhat familiar with the format, and so what we really liked about it is shown on this slide. So this is a reproducibility experiment done in my lab where we took samples with 5 different fixation times. 3 different people took 3 different samples and walked it through the HTG process and produced, in this case, a targeted gene expression assay. And what we're comparing, each dot's a gene and from each sample, and showing really great reproducibility from operator 1 to operator 2, operator 2, operator 3, and 1 and 3. And so we're really comfortable with the level and the quality of the data that we got with the base technology. But what we wanted to do is continue to move farther, is because targeted panel in this instance is fantastic, but pipelines move, questions evolve, and we really want to be able to learn more. Small samples, we only get 1 shot to test them oftentimes. So we want to have a test that's more expansive than just a target. In this case, was about 150 gene panels. We wanted to ask, more elaborate questions, but at the same time, try to strike the balance to retain the fast turnaround time where we get data in under a week, and deliver data. And that way, we can deliver data when it can be impactful, and also keep the key elements that we liked about the technology, and that's the reproducibility, the sensitivity to process small and damaged samples. So we, and this is really critical, because especially that sensitivity is dealing with patient samples that we get. There's been a trend, of course, in the field that biopsies are getting smaller. Techniques to sample tumor, diagnose tumor, are getting less invasive, moving all the way from surgical resection on one end to needle core biopsies, and then finally liquid biopsies. And so we really need to ensure that the technologies keep pace with the diagnostic procedures that are being done to extract samples out of the patients. And as these samples get smaller, they become more precious, and so all the reason to continue to use technologies that are more expansive in their coverage. And so we and certainly others have looked at how, and many different techniques in the field, and how can we understand how to get gene expression across the transcriptome and do it faithfully and rapidly. And so here's an example table that we have of the HTG transcriptome, RNA-Seq vendor 1, RNA-Seq vendor 2. And so what we do is really try to compare technology as best we can, apples to apples, and say, okay, what's the input requirement needed? And in this case, the tissue requirement for the transcriptome was 11 millimeters squared. From other RNA-Seq approaches, it's north of that, maybe 50 millimeters squared or higher than that. If one wants to isolate RNA, what kind of sensitivity are we talking about for input? And again, what we see is, a 35 to 50 nanogram input of RNA and turnaround time and other metrics. And so overlaid on the right side is about 300 cases that my group extracted RNA from in a non-small cell setting in this squamous cancer. And really to understand what yield is typical that we get out of patients with stage 4 lung cancer. And so you really see sort of a distribution plot where, maybe 30% of cases or so have less than 100 nanograms. And so using that sort of metric, we really wanted to try to see if we could get a method that is encompassing of about 90% of lung cancer above. And so in that case, we really want to be down and closer to the 30 nanograms range, and certainly south of 100 nanograms. So with that sort of input requirement, that led us to try the HTG technology when it became available. And as their technology moved to a whole transcriptome test, some features that had changed were really the coverage. Many of the features that stayed the same were things that we liked, and that is sort of this plug and play method, where it was largely the same steps in the lab for our personnel who run the test. Now just had a different container largely to do library prep with, but the upfront sort of speed and relative user friendly from an RNA-Seq point of view. Method stayed the same, where sample prep goes into the library prep, amplification, and all the downstream steps. So we still did like the plug and play method that didn't change. And then we also liked the built-in QC steps to ensure that we're able to get data that should be representative of what we'd expect. And so this is what the technology offered, and so we tried to demo it out to see if we were comfortable with it. And so another feature that gravitated us towards this is the data output, is because it takes quite a long time, as I think we're all aware, to do informatic processing of large data runs. But here the data come out of the machine analysis ready, and in either sort of here's an RUO format in the middle here of really what the gene expression values are for each gene in each sample, and then there's a data analysis suite that's available. And so this really made it very consumable on the back end, so that it really simplifies the informatic process. So that was another feature we really liked about and have liked and continue to like about our experience with this technology. So with those features in mind, I want to also discuss about what it is, what it isn't, and how it's different than traditional RNA-Seq. So in a cell, sort of at the left side in the middle, Of course, we have mRNA. And how the HTG EdgeSeq works is it's a 50 base pair probe, one for each gene, that anneals, aligns, you digest, and then the steps of amplification and RNA-Seq to basically count. And the number of times a probe is counted is reflective of the expression level. So it is one probe per mRNA. And so your value is restricted by where that probe binds. Traditional RNA-Seq, of course, you extract your RNA, you can prepare your library, sequence it, then you align it to genome. That's why it's informatically heavy. And then you can do your analysis. And so it enables you to do things like look at transcript values and things like that, but it's also just a little bit more complicated from an informatic point of view, but it is a little bit versatile from that point of view in terms of if you really care about a transcript level or an isoform level. Another thing that's really, though, critical for us working in the FFPE space is that probe length. It is because you really want to keep it on as short as possible, and so the 50 base pair probe from the HTG technology is really ideal for FFPE tissues. That really makes it really robust on degraded samples. And so the two technologies do work a little bit different, even though they're used sequencer on the back end. And so with that, another comparison here is molecularly, how are they similar or distinct from other technologies in terms of RNA-Seq? We also thought this comparison is worthwhile, is to look at the the HTG Transcriptome Panel and look at its complexity or lack thereof compared to other well-established panels in the field. Is because, one thing we were concerned about is it's a transcriptome level coverage. How will this really be too complicated over time? And so what we did is we compared it to the Illumina TrueSight Oncology 500, which is run in many CLIA labs, across the country, as well as just a different Illumina panel that, again, is also widely used in different settings. And so what we see is, in terms of although we're getting transcriptome level coverage from the HTG panel, the input material is on par or less than these other sort of well-established panels. The turnaround time is similar, if not better in some situations, and really the library, the sequencing technology and power you need is also less, and so it's really then it kind of just highlights that yes, you're able to get high level coverage across a transcriptome, but this technology is no more complicated and much simpler in many regards than something like the TSO 500, and so and easier to run in a laboratory setting, certainly. So we also thought that was a really unique sort of feature of this technology. And so, finally, what we also want to point out here is not only are we getting a lot of data and getting a lot of data rapidly, but here's 4 different biomarkers compared to what we would, loosely call a gold standard, and that's RT-PCR. And so not only are we getting data, but the data are really high quality. And you see great correlation across, many different log folds in many different biomarkers to something like RT-PCR. And so again, it's not really all that complicated compared to sequencing panels. It's much simpler, and we're able to get transcriptomes, and the data produced is really, really quite good. And so this is a unigene approach, and obviously with RT-PCR compared to a transcriptome panel, we, as I started at the talk, use RNA signatures quite often. And that's really why we wanted to implement a panel like this that has that breadth of coverage. so that we can apply these signatures and understand biology just beyond a single gene. And so here's data with their statistical colleagues. Sort of on the left, it's just looking at principal component analysis of 3 different runs, and these are all clinical samples, FFPE taken from patients. And the bottom line is we see no batch effect. And so what that enables us to do is build really robust classifiers. So that way when we get new patient samples in the door tomorrow, the day after, we can assign them with really high fidelity into 2 bins based on biology. And so on the right side is just sort of a heat map of one of the classifiers from a disease we've developed. And then, again, as patients enroll into our studies, we're able to understand their biology at a deep level and do it really robustly. And as I said previously, in time frames that enable us to make decisions. And so that's really been useful during the context of our clinical studies. And one of the other case studies here is really a sample that came from one of, again, our non-small cell studies, and this is a needle biopsy. And you can see it's not very big, and it's kind of chopped up in the middle. And this was a patient that had, let's just say, extreme interest to us from the response in the study. And so after doing IHC for the target, we only had 6 slides left. And so we asked some of our RNA-Seq vendors, how many slides do you need? And they said at least 10 to even make a go at this. And so we took 3 slides, put it into the transcriptome assay, and it worked. We got really high quality transcriptome from this sample. And so this, really does highlight, real life setting where we don't get to choose, what samples are of interest and which ones have the most material. And so when we get them, being able to turn around a result is really quite remarkable in a setting like this. And what it enables us to do is to evaluate several different hypotheses related to patient outcome and come up with one that was in our top 2 or 3 hypotheses that we wanted to evaluate, but we would have simply exhausted the tissue if we had to go through one by one and ask these questions. And so being able to do it really holistically on just a few slides really made a difference on understanding this patient and therefore our drug in a situation we probably would have been unable to otherwise, just given the sensitivity of the technology. So we thought that was a really, really great case that early on, a few months ago, really showed a lot of power for this type of approach. And so as I said, I've talked most throughout this talk about clinical samples is because that's really where most of my research relies upon. However, we've started to then move backwards into our research in terms of non-clinical approaches. So we always are interested in what engenders drug resistance. And so our internal teams generate these types of models and that are resistant to either our therapies, other therapies. And we want to understand the mechanisms by which that occurs. And so, historically, again, we send out for RNA-Seq, we wait 2 to 3 months and we get the data back. And then we design the next studies. Well, now what we started to do is shift that a little bit and say we have patient samples profiled using this technology. We have a library of, if we know target 1 and target 1's expression in patients with lung cancer, we can take these novel preclinical models, profile them using the same technology, and again with this transcriptome-level coverage, and understand then what exactly that patient tumor and that preclinical model's expression is, in concert. And then the rapid data generation that comes back, that we're able to get these results within a week instead of 2 to 3 months. And so what you can immediately appreciate is the ability to then move on to the next experiment, and the next experiment is much cleaner, much simpler, and we get data and can make decisions instead of just waiting for the data to be produced. And then when that data comes back again, we can put it and align it right next to patient samples and have the guesswork or the informatics taken out of where that sample sits on a spectrum of patients with cancer. And so this really is turned out to be a useful tool to add to our existing arsenal of other tests we may do. And so it's really been an interesting technology to bring into the lab and evaluate. And so I think at that point I'd like to turn it over to Miriam to talk a little bit about the technology in more detail.
Thank you, Pete, for this wonderful overview and for providing some examples of how you're using the HTG technology to advance your research at Abbvie. Over the next few slides, I would like to provide a quick of what HTG is all about and do a deep dive into our technology so that people really understand what it's all about. I will start with the mission of HTG. What we're focused on here is really enabling researchers and clinicians to harness the power of RNA. We are an RNA company, a company that develops tools for measuring RNA profiling. And we see a lot of potential of RNA in the precision medicine space. Obviously, DNA and proteins have been key components of the precision medicine paradigm. We believe there is a lot of potential for RNA because RNA, and messenger RNA specifically, reflects what's currently happening in the cell, which genes are being transcribed from DNA and subsequently translated into proteins. And we believe that this can really help making precision medicine even more precise. The technology that we've developed to profile RNA is what we call the EdgeSeq workflow. Pete already talked about this at a high level, and it starts with preparation. One of the factors that differentiates our technology from other RNA profiling technologies is that the EdgeSeq workflow is extraction-free. This has several advantages. It means you use less sample, but you also eliminate some of the biases that may be associated with the process of extracting RNA, or fragmenting it, or cDNA synthesis. We worked with a variety of sample types, tissue, both FFPE and fresh frozen, blood-based sample types, cultured cells, essentially anything that has RNA in it, we can process. Once the RNA is released from the biological material and stabilized, we proceed to the library preparation step. On the next slide, I'll dive a little deeper into how the technology actually functions. But once the library preparation process is complete, we then follow the standard workflows or steps in the workflow that you would expect to see and are used to from RNA-Seq. So we amplify the library, we clean it up, we quantify the library so that we can assemble a balanced library pool, and then we proceed to sequencing. The other benefit of our technology is that it's sequencer agnostic. So we can just use specific sequencing adapters and have shown a high degree of reproducibility and accuracy across a variety of sequencing platforms. On the back end, for all the reasons that we've discussed, the data analysis is quite a bit simpler as well. And not only is the output analysis ready, we've also developed a data analysis suite to help our customers, especially those that do not have in-house pipelines for expression profiling already developed. This tool really allows them to visualize and start analyzing the data as soon as it comes off of the sequencer. The other capability that we've added relatively recently were some signatures that basically take the raw expression profiling data to the next level. And a couple examples that I wanted to mention is the cell of origin signature for diffuse large B-cell lymphomas, or we've also developed a set of immunophenotyping signatures that basically estimate the relative quantity of different immune and stromal cells in the tumor microenvironments. At its core, the HTG chemistry really is a nuclease protection assay. Pete mentioned this in his presentation. For every target of interest, we develop a nuclease protection probe. This is a short fragment of RNA that has the target-specific sequence in the middle, shown in orange, and also two flanking sequences on either side. When this probe hybridizes to the target RNA, it forms a paraduplex, and there are another DNA sequences called the wingmen that basically protect the wings from a degradation. Because this is a nuclease protection assay, the next step is S1 nuclease digestion. What this step accomplishes is that it basically removes all single-stranded RNA and DNA, and what you're left with is a stoichiometric ratio of the target RNA and these nuclease protection probes. One thing that I want to highlight is that in the downstream steps, we don't actually work with the target RNA itself. We're using the nuclease protection probes as a surrogate or a proxy. This allows us to process the sample much more effectively and efficiently. So in the next steps, we basically use the wings as landing sites for molecular barcodes and sequencing adapters, and then the resulting PCR products are then cleaned up, quantified, pooled, and proceed to sequencing. The HTG technology is something that we've been working on over the last decade. It started with the launch of the whole transcriptome microRNA panel back in 2014. And as you see on this slide, over the years, we've developed a number of targeted panels, especially in the oncology space, but also later on in the autoimmune space. In addition to these commercially available panels, we've developed a number of companion diagnostic panels that have been transferred successfully to CLIA labs. What we've realized that no matter how hard we try to curate the gene content for these smaller targeted panels, there are always some genes that our customers and collaborators are missing. So we decided to develop a whole transcriptome messenger RNA panel, and I will use the next few slides to describe the features and share some data on the performance of the panel. But at a high level, there are three key differentiating factors. Increased sensitivity, as Pete mentioned, our technology uses quite a bit less sample than other RNA profiling technologies, and it maintains effectiveness with degraded samples. And I do actually have some data to that effect to share with you today. The workflow is simplified, which translates to simplified bioinformatics and also a shorter turnaround time. There's really nothing customers are giving up when it comes to performance. We've shown excellent concordance with RNA-Seq, and we'll look at some data on the following slides. I will go through some of the features of the panel on this slide. It includes essentially all protein coding genes, almost 20,000. And you can get this whole transcriptome profile from a few millimeters squared of FFPE tissue, and we also support extract and packaging of sample types. We also support essentially all production scale sequencers from Illumina, but as I mentioned before, this technology is expandable to other sequencing platforms as well. And you can do all that in 3 days or less. The next few slides cover some of the performance characteristics and data of the whole transcriptome panel. And the first one that I want to focus on is repeatability. Many of our customers test samples in a single replicate, so it is important that they have confidence in the single data set that they generate. And what you see on this slide is repeatability of technical replicates tested from 30 different sample types. FFPE on the left-hand side, PAX gene in the middle, and extracted RNA on the right-hand side. And as you can see, repeatability is really excellent across all of these sample types with Pearson correlation coefficient north of 0.9, which suggests a high level of repeatability. Of course, repeatability and precision are only one side of the equation. Accuracy is the other. So we've put a lot of effort into proving that our technology is highly accurate. And the obvious way to do that is to compare to a gold standard. But before we decided to do that, we really wanted to use an independent way of proving accuracy. And that is particularly important if the gold standard that you're comparing to is not so golden. The way we did this in this experiment was using these exogenous RNA transcripts that were spiked into the sample at various concentrations. These transcripts span a concentration range of almost 1 millionfold, and as you can see, there is a very good correlation between the amount of spiked material that's shown on the x-axis and the signal coming from the assay on the y-axis. The Pearson correlation coefficients are really high, and not only is this relationship demonstrating a high degree of correlation, it's actually linear across a very broad dynamic range, approximately 20 orders of magnitude on a log 2 scale. So this really gave us confidence that the panel was highly accurate across a very broad dynamic range. Of course, a lot of our customers come from RNA-Seq, and they want to be confident that the data they've generated so far can potentially be combined or leveraged for their downstream studies with our technology. And what we did in this experiment is comparing the results of differential expression analysis between the transcriptome panel and RNA-Seq. The way this experiment was executed is 2 samples, breast cancer and melanoma, were tested on each of the 2 technologies, and then we used a differential expression analysis to determine log-fold changes for each gene in the panel. What you see on this plot is basically a comparison of the log-fold changes from the differential expression analysis. And again, we see a very high level of concordance between the HTG panel and RNA-Seq. One last data point that I would like to share is a comparison to RNA-Seq with samples of various ages. And this is important because one of the distinct advantages of our technology, as Pete mentioned, is the ability to work with partially degraded samples. So we used a cohort of FFPE tissue blocks that were collected relatively recently, blocks that were 10 years old or older. And we tested these samples both on the transcriptome panel called HTP and RNA-Seq. In all instances except one, the HTP panel required only a single 5-micron slide of the tissue, whereas RNA-Seq required anywhere from 4 to 8. The overall pass rate for our technology was 100% in this cohort, whereas in RNA-Seq only 75% of the samples generated actionable data. And as a matter of fact, this number dropped to 63% when we only looked at the subset of the samples that were older than 10 years old. So I think this really highlights the ability and robustness of the technology to work with small or partially degraded low quality samples. So there are a lot of benefits, and just to sum it all up, not all transcriptome profiling tools are created equal. RNA-Seq is an extremely powerful technology, but it requires a lot of high quality sorting material. Our technology, on the other hand, can work with much smaller amounts of sample. The workflow is simplified because it simply skips some of the steps that are typical for RNA-Seq without sacrificing the quality of the data coming out of the technology. And the assay success rate is also very high. As we see, it was about 96%, and these are data from our commercial laboratory that sees thousands and thousands of samples every year. So I realized this was only a quick overview of the technology, and I will now hand it back over to Pete for his closing remarks. Thank you.
Great, thank you, and thank you everyone for the time, for listening. And I know I'll have a Q&A after this, but I also wanted to thank many of the folks in my lab here at Abbvie, and just in Abbvie in general, Precision Medicine Group. Lisa, Lane, and I have been really strong internal supporters here to either generate a lot of the data, get support for a lot of the resources, and then some of the experiments we showed today really were the work of numerous clinical trials. Where we're putting many different samples together to come up with these types of conclusions, and project teams, clinical sites that all went into the clinical programs, and then of course the patients and the family who donated the material to try to help cancer research get better in the future for them and other patients. So and again, thank you for your time. Happy to ask and answer questions.
Yeah, fantastic. Well, thank you, Pete and Marion, for the really great presentations. All right, so let's jump right in. And Pete, we can start with a question for you. So this attendee has said, I often face internal pushback when I try to introduce a new technology to my laboratory. People are apprehensive about branching out from a comfort zone and trying something new and unfamiliar. What would be your advice for me when I have these conversations with my team?
Well, sure, that's a great question. And if I may, let me go to Marion's slide 24, because I think this really captures, the workflow, for example, nicely. And that is, this is basically just RNA-Seq improved upon. And you kind of think of 4 steps. How do you make sample? How do you make a library? How do you do your sequencing? And how do you do your analysis? And so really take your sample and scrape it into a tube has really simplified that. A semi-automated library prep, has simplified that part. The sequencing, again, if you already have a sequencer, you're familiar with it, that's really, well established, and they would be familiar with it. And then the data analysis. So of the 4 steps, it's really quite analogous to probably what they're already used to, just simplified. And so I don't know, Marion, if you have that's my take. I don't know if you have anything to expand upon that.
I think that was a very comprehensive answer. The only thing I would add to that is what attracts a lot of our customers to HTG is the ability to work with small and partially degraded samples, and that's particularly true for situations where you're dipping into your sample archive. Oftentimes, customers are scraping the bottom of the barrel, and being able to leverage some old clinical trial samples and basically get the entire human transcriptomic profile can be extremely informative.
stated.
Yeah, some great points. All right, so the next question, and Marion, I'll ask you to lead this one. So HTG was launched about 10 months ago. What was the main motivator behind developing such a product?
Yeah, I think this talks about HTP, which is our HTG Transcriptome Panel. That is our latest product. And as I mentioned on one of the previous slides, HTG historically was heavily focused on these smaller targeted panels focused on a specific application or set of applications. And what we've realized over the years is that there's invariably gene content that is missing. It also limits our customers to a specific disease state or disease areas. So what we've seen in the field is the trend towards more content, not less. And what we were able to accomplish with HTP is to be able to basically expand the opportunities or potential applications of the assay essentially all across all disease areas, and basically provide the same benefits of our technology with a much greater gene cover.
Yeah, excellent. All right, here's a good question, and I think maybe actually both of you could probably answer this one. So in which areas of research or therapeutics do you anticipate RNA sequencing and transcriptome profiling to become more mainstream in the upcoming years? Pete, maybe we can start with your answer for this one.
Sure. No, that's a great question. I think I also see that there was somebody, surprised about tumor types and situations that don't have actionable driver mutations. And a great, fantastic example of that are things like small cell lung cancer. You look at non-small cell, it's well-defined EGFR mutations, RAS, ALK. Even within a non-small cell, then you have about 50% of patients with an actionable driver. Small cell, you just don't see that. Or at least we haven't found the drivers or can't drug them yet, or things like that. So certainly, I think there are diseases like that are really well captured. I mean, triple-negative breast cancer is another one, by expression profiling to understand the biology. And then from a therapeutic perspective, there's clearly IO, and IO tends to be beyond PD-L1 expression. Pretty complicated signatures. Interferon gamma, things like that. So understanding response to IO therapies, which are not often well modeled by univariate responses. And then finally, I think in the drug development space, going back to that expression mantra, antibody drug conjugates. Daiichi has done phenomenally well with antibody drug conjugates targeting overexpression. We have some drugs in the pipeline certainly for that. And have presented recent data on those, and so really where we're trying to move away from the drivers are there, and we have good actionable biomarkers hitting that, but that's even in lung cancer, non-small cell lung cancer, only half the population. And so trying to come up with therapies, be it antibody drug conjugates or other, that really target overexpression, and then that's probably a lot of times where the field's moving.
Yeah, great answer. Marion, anything to add to that?
I will just add that besides oncology, which is a very busy field with lots of potential, we've seen a lot of great work in the areas of diabetes and neurodegenerative diseases. I'm trying to think if there's anything else. Cardiovascular. This may not be so much with messenger RNA, but with microRNA and other circulating small RNAs. So there are great applications and good potential across a variety of disease states.
Yeah, fantastic. All right, another interesting question here. Can we apply this on single cell analysis efficiently? So what would the sensitivity be for for this analysis? I'm not sure I fully understand the question, but Marion or Pete, maybe you'll get that.
I think I can decipher that. So we technically do not offer single cell gene expression profiling, but there's really nothing preventing us from doing that. The way we've optimized the technology is really for maximum signal across human transcripts. And whenever you move to single cell analysis, you're giving up some of that sensitivity. But we've done some proof of concept work with a small number of cells or laser capture microdissection. So the technology definitely is capable of going way down and sampling, but you're going to pay for it with sensitivity.
Fantastic. All right. I think we have time for maybe 2 more short questions. Have you used HTG for microRNA exosome? I'm not sure who would be best to address that one, but
Probably Marion.
I'll take that one, and maybe I'll combine it with the next question because they're actually related. This would be the second question is, can you expand on the use of blood cells versus plasma? The technology obviously supports a variety of sample types, and in our commercial laboratory, we work with blood-based sample types very routinely, whether it's plasma, serum, hex gene, or sorted PBMCs. We've done all of them very successfully. When it comes to exosomes specifically, it is very much possible, and we actually do that on a semi-regular basis. Our customers oftentimes approach us about exosomic profiling, but as many of you know, especially those who are active in this field, the process of enrichment for exosomes can be extremely variable. So if you have that part of the process mastered, our technology is perfectly capable of analyzing those purified exosomes and generating some microRNA commercial profiles.
fantastic. Yeah, Pete, anything to add to that?
No, that was well covered. Thank you.
All right, fantastic. I think in the interest of time, we'll maybe have one last question here. Looking for one here. All right, there's one here about the relative cost for the HTG technique. Pete or Marion, can you maybe elaborate a bit on that as the last question here?
Sure. I mean, I think I can take a crack, quickly as sort of an end user. And again, I think about this as, you need to start with a sample and then through your data analysis. And so, isolating RNA versus not, there's a cost savings there. And then that data analysis piece, and that's, what I stressed a few times throughout the talk is some vendors who do RNA-Seq, you will pay a lot of money for. That analysis piece more so than the transcriptomic profiling. And if you do that internally, then there's an overhead to that as well because you need to hire informaticians. So it's actually, but even all that withstanding, quite comparable, or cheaper than some of the full service RNA providers. But
yeah, fantastic, great answer. All right, well, I think with that, will have been the last question. So yeah, again, Marion and Pete, thanks so much for all your great insights today. It's been a real pleasure having you with us.
Great, thank you.
Thank you.
Yeah, big thanks also to the audience here for joining us. And of course, we'd like to thank HTG for helping to sponsor the event. And so in closing, we hope you enjoyed this Scientist.com webinar, and we look forward to having you with us again soon. Have a great day, everyone.